Taylor West.
And our project is on cryopreservation of plant cells. And so cryopology is the study of life at low temperatures. And although freezing is lethal to most living systems, cryopology can allow long-term preservation of tissues and organs. And this is the purpose of our experiment, to produce stable conditions in which life can be preserved.
The process of stabilizing biological materials of cryogenic temperatures is cryopreservation, a practical application of cryobiology. The process of cryopreservation involves preserving living cells and tissues at very low temperatures and then gradually thawing them out to room temperature. The purpose of cryopreservation is to produce stable conditions in which life can be preserved. When the living cells and tissues are frozen at negative 80 degrees Celsius for 7 days, the water in the cells are converted to ice, which causes all cellular metabolism to cease. The cells are pre-treated with DMSO and glycerol. These are used to protect the cells by making them less prone to puncturing and interrupting vivification. Vivification is the formation of sharp ice crystals when the water in the cells become frozen. The process of this research is to conduct viable tests to determine the percent of recovered living cells. Although used intensely in food preservation, the subfreezing storage of tissues and organs has also found application in medicine. As stated before, cryobiology is important in the medical field. Cryopreservation is used today for fertility treatment, transfer of human organs, and long-term storage of biological specimens. Embryo cryopreservation is often used along with IVF programs. During the embryo production process, many embryos are often made. Most of these embryos are placed within the woman's uterus, but some of them are preserved through cryopreservation. This is a vital step of IVF programs because if the first implants are unsuccessful, the frozen embryos are thawed out and prepared for use. This is why cryopreservation is essential because it provides the patients with more opportunities to become pregnant.
So in order to prevent this cell death during preservation, we must use cryoprotectants. And there are various reasons why cells die in freezing temperatures. It could be from rapid cellular ice formation, cell shrinking, and osmotic oscillation. So cryoprotectants begin to dissolve in the preservation of the solution, and they work by lowering the melting point of the solution. And so intracellular cryoprotectants begin to penetrate inside the cell, and this prevents the formation of ice crystals inside the cell. And commonly used intracellular cryoprotectants are glycerol, DMSO, and propylene glycol. The two that are used in this experiment is glycerol and DMSO. And they are advantageous for this project because glycerol forms hydrogen bonds with water, making it very hard for crystallization. And then DMSO, along with lowering the melting point, is also very cost efficient and has a low cytotoxicity. So now we're going to move to the materials and methods section. So we began our research by extracting callus from tobacco plants. And we excised a small portion of the callus and placed it into a flask. And then we filled with liquid MS. And then we let the callus incubate for 48 hours. And then using a sterile cheesecloth, we separated the callus from the media. And then we obtained 3 separate tubes. And so in one tube, we had 5% glycerol, 0.5% DMSO, and we used one as a control. And these were also filled with the same amount of liquid MS. And so these tubes were placed into the freezer. These tubes were placed into the freezer at negative 80 degrees Celsius for 48 hours. And then after removing from the freezer, these tubes were placed into an ice bath for 5 hours to allow them to gradually thaw. And then after reaching room temperature, 1 meal of each tube was centrifuged for 3 minutes at 10,000 RPM to separate the supernatant and pellet. The supernatant was then discarded, and the pellet containing the cells was extracted from each tube and smeared onto the appropriate section. So these are the 3 tubes. This is after they were frozen with the cells inside that we extracted from the callus. And then that's them gradually thawing in the ice bath. And then centrifuging in the very last picture, we just took one medium plate, we divided it into 3 sections, and then we labeled them with the glycerol, the DMSO, and the control. And so then we smeared it onto the plate, and then we're incubating it for 2 to 3 weeks, and to see if we have any cellular growth.
So we began this research about 2 weeks ago, so we don't have any results right now. As you can see on our plate, we don't have any growth. We do expect there to be cellular growth on the plates. So as right now, there is no clear growth on our plates because it takes 6 hours for eukaryotic cells to divide, which is why we do not have any results at this time. We are predicting that growth will occur and proving that our cells survive cryopreservation. If there is no growth, we will continue our research with new methods. But if there is growth, we will begin to do our research with cryopreservation in different ways because this is such a vast, wide open field. There are various research ideas to explore. And that's all. Thank you.
Questions? I've actually got one, which is, you listed pretty quickly 3 ways that cells could die, in the frozen state there. And one of them was ice crystals, and you said 2 others very fast.
Let's see, it was the osmotic oscillation, that was one.
Okay, now what is that?
Off the top of my head, I guess it's just osmotic pressure that the cells need to maintain
From such a
pressure.
You have to not be able to thaw them out at a decent time period. You can't just bring them out of the freezer and just, because that'll rupture the cells from freezing. Yeah, from freezing to thaw out so quickly.
Yeah. And then the other one was cell shrinking.
Okay.
Any more questions?